What's up, y'all? Professor Hurley here, and welcome to another episode of Purple Bear Biology. In this episode, we are going to cover the mind-blowing concept of electrophysiology. Neurons throughout your body are currently sending electrical signals about what you're experiencing and how our bodies should react to maintain homeostasis. Electrophysiology allows us to understand and monitor these signals. This video will build off of our last conversation that covered structure of the neuron. If you missed it, I'll put a link for it below. No doubt you have probably heard and seen the echocardiogram from just a moment ago. But what does this actually monitor? Well, it follows the electrical stimulations of the heart through contraction. This can actually be done with neurons throughout our bodies as well. Using really small conductive materials, we can measure the voltage inside of a neuron cell, and when this voltage changes, we know the neuron is sending an electrical signal. That's really cool, right? But how does the cell change the charge and send the signal? Well, I thought you would never ask. Okay, so let's jump right into it. Recall that we have four basic parts of a neuron: the dendrites, the soma, the axon, and the synapse. Dendrites will act as receptors of signals from other neurons. These signals can either be excitatory, telling the neuron to send an electrical signal down the chain, or inhibitory, suppressing signaling transmission. These small signals can occur in the form of electrical differences and are called local potentials. Recall from our conversation of the neuromuscular junction that the inside of the cell is negative at rest and positive when excited. Typically, a nerve cell will have a resting membrane potential set around minus 70 millivolts. An excitatory local potential would raise the membrane voltage to a more positive value, while an inhibitory potential would make the membrane more negative. These local potentials are summed up at a junction between the soma and the axon, called the axon hillock. If enough positive charges accumulate and the cell's membrane reaches a value called threshold, then voltage-gated sodium channels open and allow the sodium to flow into the cell, resulting in an electrical message being sent down the axon. This positive electrical signal is referred to as an action potential. It is actually really important not to mix up action potentials and local potentials. Action potentials are kind of an all-or-nothing thing. Either you reach threshold and send an electrical signal, or you don't. Also, there's no degree to an action potential. It's either there is an action potential, or there's not. There are no strong action potentials or weak action potentials. In contrast, local potentials are said to be graded. This means that they vary in degrees of excitatory and inhibitory signals. Stronger local potentials can have stronger effects on a neuron's ability to send signals. In addition to these differences, local potentials are also just localized, meaning they stay in one general area. Whereas when we talk about action potentials, they tend to send signals over long distances relative to local potentials. And then lastly, by default, local potentials can cancel each other out. One excitatory signal could be canceled by an inhibitory signal and never result in a neurological action potential. However, once an action potential is triggered, it cannot be stopped or reversed, meaning that once we do trigger a neurological stimulus in the form of an action potential, there's no pulling back. It's like a bullet out of a gun. Once the trigger is pulled, or once the threshold is met, the bullet leaves, or the action potential leaves the axon hillock and travels down the axon. Okay, so let's break this process down a little more and take a closer look at how action potentials get passed down the axon. So as we saw before, threshold is reached when excitatory local potentials raise the membrane charge. The threshold value can vary greatly depending on a neuron and the ion concentrations around the cell, but typically this falls somewhere around minus 55 millivolts. So let's take a look at all of this on a graph. On my y-axis, I have voltage, and in the case of neurons, we're talking about millivolts. Now, 0 would occur approximately right here, and then resting membrane potential is normally around minus 70 millivolts. So this would be our resting membrane potential, and is often abbreviated as RMP. Threshold is the value that has to be reached in order for an action potential to fire. Typically, this is around minus 55 millivolts. Once local potentials accumulate to minus 55, the first voltage-gated channel found at the action hillock will open and allow sodium to flow in. The rest of the steps take place from the axon down. So we're going to draw this out so that we can take a closer look at what the inside of the axon looks like and how it's changing through this process. I'm going to draw the axon like this, where this is extracellular fluid and this is intracellular fluid. The inside charge at resting membrane is negative all the way down. The outside charge at resting membrane potential is positive all the way down. As sodium enters at the action hillock, the charge increases inside of the membrane. This charge is recognized by voltage-gated sodium channels embedded into the axon. The charge change allows sodium that is in higher concentrations outside of the cell to travel in. As sodium pours into the cell, this creates a charge change. And the next sodium channel recognizes this change and allows sodium to flow in, effectively creating a positive charge. This positive charge gets passed down the membrane by opening voltage-gated sodium channels along the way. This results in a dynamic charge flip and is referred to as a depolarization event. On our graph, local potentials reach threshold and then trigger the first opening of the sodium channels, and this results in a very positive value inside of the axon. This is referred to as a depolarization event. Now, the membrane does not stay positive because this would prevent the neuron from sending another signal. So to reset the electrical charge, the cell will pump positively charged potassium ions out of the cell. As it pumps these potassium ions out of the cell, this creates a negative charge on the inside of the membrane, restoring our electrical gradient. As it does this, it causes the electrical charge to decrease back down to resting membrane potential. This is called repolarization. Sometimes too much potassium actually leaves and the charge gets lower on the inside of the membrane than resting membrane potential. It eventually will rise back up as the nerve cell restores resting membrane potential, but during this period, it's harder to send an electrical signal across the neuron, and this is referred to as hyperpolarization. Now, you may have noticed a problem with this condition. We have stored the electrical charge, but our ion concentrations are not set up to send another electrical signal. So to fix this, our cells use an additional protein called the sodium-potassium pump that pumps potassium back into the axon and sodium out of the axon. All of this happens in a fraction of a second and is occurring in billions of neurons throughout your entire nervous system. Do you remember the conversation about neuroglial cells called Schwann cells and oligodendrocyte cells that can speed up the signal conduction? How does that really work? Well, non-myelinated fibers must open voltage-gated channels all along the axon, but myelinated fibers are insulated, and the neuroglial cells maintain the electrical charge from one exposed area of the axon to the next. Essentially, I think of this like non-myelinated axons are like walking from the house to the grocery store, while myelinated axons are like skipping to the store instead. The electrical charge gets to hop along and is more rapid in these myelinated fibers. So what happens when this electrical signal reaches the synapse? Well, in most of our neurological connections, we have chemical synapses where neurotransmitters get released to transmit a signal to another neuron or to innervated tissues. But some of our neurons have electrical synapses that enable ion movement to continue electrical stimulation to the next cell. Okay, so this is all cool, but again, let's make sure that we can apply it. So let's talk about this whole process, focusing in on two primary ion concentrations that affect the electrophysiology of our nervous system: calcium and potassium. Let's look at calcium first. We have taken a look at calcium regulation in our blood before when we talked about the endocrine regulation of bone building, breakdown, and calcium reabsorption or excretion. Blood calcium levels directly affect threshold levels of our neurons as well. Hypocalcemia lowers threshold, and hypercalcemia raises threshold. So how would this affect our neuron firing? If threshold is closer to resting membrane potential, then the neuron will have an easier time firing. However, if threshold is further away from resting membrane potential, then it will be harder for the neuron to stimulate an action potential. So, hypercalcemia suppresses neurological firing, and hypocalcemia increases the likelihood of neurons firing. Now let's look at potassium. We can have high levels of potassium in the blood, referred to as hyperkalemia, and this raises resting membrane potential. We can also have low levels of potassium in the blood, referred to as hypokalemia, and it results in lowering resting membrane potential. Don't forget that the distance between resting membrane potential and threshold determines the amount of stimulation needed for an action potential to be triggered. So, hypokalemia increases the distance and makes it harder for neurons to fire, while hyperkalemia decreases the distance and makes it more likely for neurons to send signals and action potentials. Well, y'all, that's it for this episode of Purple Bear Biology. If you liked the video, be sure to click the like button and subscribe to follow along with our next conversation over the different regions and functions of our central nervous system. Thank y'all for watching, and see you next time.